Today we have the microscopy lab, and we are going to use two bacteria, Escherichia coli and Staphylococcus aureus, the mix of bacteria, the yeast, and a part of onion skin. So for that we will need 5 slides like this, and we are going to label them first. So we have the labeling surface here, and it is rough on one side and smooth on the other, so we are going to use the rough one to label our slides. So first one will be Escherichia coli. Then we have Staphylococcus aureus. Then we have bacterial mix. Then we have yeast. And finally, onion. So we start with the preparation of our samples, and we take ten microliters of water and place them on the middle or in the center of the slide. So we place it here. And then using the loop, we are going to take Escherichia coli single colony, and we are going to use the Bunsen burner so that we don't make our environment contaminated. So we take a small colony, and it is now on the loop, so we transfer it to the glass, and making circular movements, we just make this drop go a little bigger in diameter. So we usually take a 5 tenge rough area and we spread it like this. After that, we use burner to kill the bacteria. Next we do the same for Staphylococcus aureus, and again we take 10 microliters of water, place it in the center of the slide, obtain single colony of Staphylococcus aureus, and again it's on the burner. So we take one single colony and spread it in the same area as Escherichia coli. After we've done it, we again kill bacteria, and then we use the bacterial mix. In this case, we are just going to take the bacteria that were prepared before that. So this mix should include both types of bacteria. We take it and spread it as we were doing before, in the area of about 5 to 10 tenge. You should be able to spread it easily, because with the bacteria the surface tension is not the same anymore. We repeat the procedure with the yeast. So we take 10 microliters and spread it evenly on the area of about 5 tenge. Finally, we have our onion sample. It won't take a lot of time, so we will do it later. For now, we are waiting until these areas are dried out, so we can continue with the staining. So as you can see, yeast is already fine to go, and it is dried out. As you can see now, the yeast sample is already dried out, so we can heat fix it. We rapidly pass the slide through the flame 2-3 times, so 1, 2, 3, and we can place it on the slide holder for further staining steps. Okay, now you see that the sample of the Escherichia coli sample is also dried out. So we also pass the slide through the flame 2-3 times rapidly to heat fixate it. Now we have 2 samples that still are wet. Now we have Staphylococcus aureus also dried out, and we also fix it by passing the slide through the flame. Finally, we have only slide with a mix of bacteria that is almost dried out. Okay, so now we have the last slide dried out, and you don't see the drop there, so we also fix it. And now we can switch to the next step of staining, and the first step would be putting the crystal violet on top of these slides. So we will be putting about 200 milliliters. We need to cover all of these patches with the crystal violet. So drop the crystal violet on top of these patches. And we need to wait for 1 minute. Okay, now it's time, and we wash it with water. We don't need the Bunsen burner anymore, so we turn it off and close the gas. So we remove water. And place the needed amount of iodine. So we place it on the same place. We apply it for 1 minute, about 1 minute. Okay, we wash it with water. Next step would be to decolorize the samples, and to do so we use 95% ethanol. So don't overdo that. And wash it with the water. So we wash the alcohol with water so it does not over decolorize our samples. And we have final wash, final stain. Now we are going to use saffron, and we'll also apply it for 45 seconds. Now we can blow dry them. To do so, we take the tissue and gently place on it so that all of our samples are intact. And after that, we're going to use oil immersion to look these samples up.